Hello guys, welcome back. So let's start with the short notes video number 10. I hope everything from the previous videos are clear, and if you have got any doubts then feel free to comment your doubt, or you can also DM us on the Instagram account. The first organism for this video is going to be pertussis. It is a disease. Pertussis is a disease which is caused by Bordetella pertussis. So the agent for this is Bordetella pertussis. Now this disease is also known as 100-day cough, or we call it as whooping cough, because it causes a very violent cough. Okay, so it causes whooping cough, or we also call it as 100 days cough. For staining this Bordetella pertussis, we have a specific stain. So you should remember the stain for it is Bordet Gen Go stain. A very important point. Okay, please remember the stain name. And lastly, we have vaccine for the prevention or prophylaxis of this organism. So vaccine we give is a combination vaccine, which is a DTP vaccine. The full form for this is diphtheria. D stands for diphtheria, T stands for tetanus, and P stands for pertussis. Okay, so this is a combination vaccine. And we have another vaccine which is APDT. Now this stands for acellular. A stands for acellular, P is for pertussis, D is for diphtheria, and T is for tetanus. Okay, so acellular PDT I'm writing. I hope this makes sense. And we should know what is this vaccine. It is a booster vaccine given to children. Okay, booster vaccine in children. So DTP normally given to adults, given to children as well. As a booster vaccine, we also provide APDT, which is acellular pertussis, diphtheria, tetanus. Okay, it is a combination vaccine. So we know that viruses have different types of serotypes, and we try to detect the serotypes of different viruses, right? So how exactly is it possible for us to check it? Let's know some things about it. First, we know that viruses contains capsid, right? Now this capsid, it is made of proteins. And these protein acts as an antigen in our body. Our body recognizes all the proteins in us. So if there's a virus coming, so that means they have this capsid. On capsids are present the proteins. So those proteins are different from us. So our immune system is able to recognize these viruses as an antigen. And then what will our immune system do? Our immune system will start producing antibodies. So therefore, antibodies works against what? Antibodies are released against these proteins. Okay, because that is what is counted as an antigen by our immune system. Okay, therefore, to check the serotype of viruses, therefore if we want to check the serotype of viruses, then antibodies produced against proteins of capsid should be used, because different antibodies will be produced against different capsid protein, right? So we can use the antibodies produced against the proteins of capsid to check for different serotypes of viruses. Okay, now we know that immunofluorescence is a express test or express diagnostic or quick diagnostic test, right? Immunofluorescence is express diagnostic or quick diagnostic, right? But if in the option you cannot see this immunofluorescence, then what will be your second option to mark? Then if this is not present, then the best second answer would be immunoelectrophoresis. Then the answer is immunoelectrophoresis. Next, let's learn something about influenza. Now, for influenza virus research methods, it is done by two ways. One is isolation and another is serological identification. Okay, serological identification includes agglutinations and everything. Okay, antibody antigen reaction. Let's know some information about isolation. So isolation is done by infecting chicken embryos. So we infect the chicken embryos, but before infecting, we decontaminate the research material by streptomycin and penicillin. Okay, so what does it mean by isolation? Isolation is where you are infecting the chicken embryos. Okay, but before infecting them, you will be decontaminating the research material by using streptomycin and penicillin, basically antibiotics. Okay, then you should know something about the surface glycoproteins of this virus. So surface glycoproteins of this virus are two, which acts as an antigen. Okay, proteins on the viruses acts as an antigen. We just did it in the last point, right? So here the surface proteins are hemagglutinin, and second surface protein is neuraminidase. Okay, now these two acts as an antigen, and therefore it is asked immunity to what is developed in case of influenza virus is infecting a patient. So what exactly is the immunity developed to? We did it in the last point, that antibodies are created against the antigen. Now what exactly is an antigen? It is the proteins, the surface glycoproteins in case of influenza, which are these two, hemagglutinin and neuraminidase. Therefore immunity is developed against these two. It is developed against these two. Okay, I hope this is clear. Okay, now the next thing we should know, that there are some viruses which affects the sex glands. So we should know the properties of such viruses. So viruses properties that affects sex glands. So I'm listing down some of their properties. So if you see such properties, then you should be sure that it is a virus which is going to affect the sex gland. So these viruses, they replicate in chicken embryos. They have hemagglutination properties, and they form symplast in cell culture. Now symplast in viruses, it is a space where viruses reproduce and also eliminates any foreign RNA. Okay, so the characteristic for the viruses which will affect the sex glands include that they replicate in the chicken embryos, they will have hemagglutination properties, and they will form symplast when they are cultured. Now symplast is what? It is a space where the viruses will reproduce and will also eliminate any kind of foreign RNA present over there. Okay, I hope this is clear. That's all for this video. Let's meet in another video again. I hope everything is clear. Thank you.